Hi class, I hope you guys are doing great and you're ready for our last week of this course. My name is Hannah and today I'm going to talk to you all about tuberculosis. I have three objectives for this presentation. The first is to describe the taxonomy and structure of Mycobacterium tuberculosis. The second is to describe the discovery of tuberculosis, and the third is to outline the pathogenesis, epidemiology, diagnosis, treatment, and prevention of tuberculosis. So tuberculosis is caused by a bacterium called Mycobacterium tuberculosis. Here is a table of its taxonomy. One interesting thing I came across in a 2024 review by Orger and colleagues is that there are actually two very different estimates for when the Mycobacterium tuberculosis complex, or MTBC, first appeared in humans. And that Mycobacterium tuberculosis complex includes all of the similar bacteria that cause similar diseases in other organisms. The first estimate comes from modern whole genome sequencing, which places its origin at about 70,000 years ago. And that lines up with when human mitochondrial DNA started to diverge. So it's possible that TB and humans have actually been evolving side by side since early human migration out of Africa. But another study based on ancient DNA from skeletons in pre-Columbian Peru estimates that the most recent common ancestor of the MTBC appeared only about 6,000 years ago, which would mean TB didn't really take off until more recently, maybe due to population growth, animal domestication, or agriculture. So basically, there's no consensus yet on exactly when TB emerged. For the structure, tuberculosis is a rod-shaped non-motile bacterium. It's about 2 to 4 micrometers in length and 0.2 to 0.5 micrometers in width. Its cell wall is a big part of why it's so hard to kill. It's made up of 3 main layers: a plasma membrane, a cell wall, and an outer layer. And the wall is packed with lipids, which makes it super hydrophobic and helps the bacteria resist antibiotics and the immune system of the host. The peptidoglycan layer gives the cell its shape and strength, and attached to the peptidoglycan layer is an arabinogalactan layer, which helps with structure and hydrophobicity. And then on the outside, there's a thick layer of mycolic acids, which make a waxy coat that kind of acts like an armor that helps protect the bacteria. The M. tuberculosis genome is about 4.4 million base pairs long and has around 4,000 genes. One thing that stands out is the high guanine and cytosine content. Almost 2/3 of its genome is made up of guanine and cytosine, and that affects how the DNA folds and even the types of proteins it makes. There's also a lot of repetitive elements like insertion sequences and multi-gene families, and these help the bacterium adapt and evolve in different environments, especially when it's inside of the host and responding to the host's immunity. A large portion of the genome is dedicated to lipid metabolism, like breaking down and building fats, which makes sense because of how lipid heavy the cell wall is. And then finally, it includes many glycine-rich proteins, which may help the bacteria change its surface proteins to help it evade the immune system. And together, all of these features make tuberculosis such a persistent pathogen. So a little bit about the history and the discovery. Before Robert Koch identified M. tuberculosis in 1882, Tuberculosis was already widespread and very deadly, but people didn't know what caused it. It was often called consumption, and it carried a strong social stigma. Evidence of TB goes back thousands of years. We've seen signs of it in Egyptian mummies from around 2400 BC. It's also been described in ancient texts from India, China, and Greece. Hippocrates noted how often it killed young adults in ancient Greece. In the Middle Ages, a form called scrofula affected lymph nodes in the neck. People called it the king's evil and believed that a touch from a royal could actually cure it. In 1720, Benjamin Martin suggested TB might be infectious, and then by the time of the Industrial Revolution, TB had exploded in crowded European cities, especially during the Industrial Revolution. It wasn't until Koch came along in 1882 that we had solid proof. He isolated the bacterium, grew it in culture, and showed it caused the disease. His work didn't just explain tuberculosis, it helped establish the entire field of germ theory. And fun fact, he also discovered the bacterium behind anthrax. A TB infection starts when someone inhales airborne particles, probably from someone with active TB. The bacteria reach the lungs and get taken up by alveolar macrophages, but instead of being destroyed, they start to multiply inside of the macrophages. And at this point, the infection becomes latent. The person doesn't really have any symptoms and they can't spread it, but the bacteria are still there. And then if their immune system weakens for some reason, maybe age, some other disease, the TB can reactivate. The immune system fights back with both innate and adaptive responses. Macrophages and dendritic cells recognize the bacteria and trigger inflammation. And then later, helper T cells and interferon gamma help activate macrophages to contain the infection. But M. tuberculosis is sneaky. It can hide inside of macrophages and promote granuloma formation. And granulomas, that's a hard word for me, help keep the infection contained, but they also provide a place for the bacteria to hide. According to the World Health Organization's report in 2024, in 2023, there were about 10.8 million people worldwide that had tuberculosis, which is roughly 134 cases per 100,000 people. Around 6.1% of cases were people living with HIV. Just 8 countries accounted for over 2/3 of global cases: India, Indonesia, China, the Philippines, Pakistan, Nigeria, Bangladesh, and the Democratic Republic of Congo all had the most cases. Men made up the majority of cases at 55%, women at 33%, and then children under 15 at 12%. There are 2 different treatments for TB depending on if it is drug resistant or not. So if you have drug susceptible TB, which means it can be killed with standard antibiotics, the treatment involves taking multiple medications for several months to make sure that all the bacteria are killed and to prevent the disease from coming back or becoming resistant to drugs. So the most common medicines are isoniazid, rifampin, pyrazinamide, and ethambutol. I've been working on those pronunciations. In drug-resistant TB, the standard drug treatment usually takes at least 6 months, and the most common medicines are bedaquiline, protamine. In people with drug-resistant TB, it usually takes at least 6 months to treat, and the most common medications are bedaquiline, protamine, and linezolid. Bedaquiline works by blocking ATP synthase, which the bacteria need to make energy, and so if you can't make energy, you can't survive. Protamine works by stopping mycolic acid synthesis, and if you'll remember from a few slides ago, that mycolic acids are a big part of the cell wall, and it also releases nitric oxide, which helps kill the bacteria. And then linezolid is able to block protein production in the bacteria, which kills them. Preventing tuberculosis includes a few key strategies. The World Health Organization recommends TB preventative treatment for people living with HIV and for people who have other people in their household who already have TB or are a part of another high-risk group. TB preventative treatment often includes screening programs, which can help identify and treat TB earlier, which helps reduce the spread of the disease and improves the outcome of the people who are now infected. And then there is also a vaccine for TB, and it's usually only given to children, or in some other rare cases, some people can get it, like sometimes nurses if they work with people who have TB a lot. Finally, here are my references. They are not the prettiest, but they're in there. Let me know what questions you have, and I look forward to seeing all of your presentations too. Have a good week.